in this series I try to explain my PhD thesis in shortish videos. So let's get into chapter 1, the multi-omic delineation of cytokine-induced endothelial inflammatory states. To preface this, endothelial cells in the bloodstream need to be able to respond to a whole bunch of different situations, like coagulation, inflammation, immune cell transmigration. To deal with all these situations and have the correct response, they need to have the ability to react to a whole range of different stimuli. And in this paper, we focus on the inflammation and in specific cytokines and chemokines. So the first question we had was, to which cytokines or chemokines does the endothelial cell actually respond? And to do this, we got a panel of 93 different cytokines and chemokines and stimulated our endothelial cells with those. So the results you can see here in this bar graph, where you can see all the different cytokines ranked on how much they induce a proteomic response in our endothelial cells. So the highest response inducer in this case is TNF-alpha, which we know quite well. The second highest that has a different response is interferon gamma. If I'm completely honest, I'm not sure what led to us combining them, but what we saw is that when you combine TNF-alpha and interferon gamma together, then you get a third unique response that is not just the combination of both, but also an added sort of synergetic response of both cytokines. So now we want to understand the molecular pathways of this response in detail of all these three different inflammation responses. And to do that, we use the multi-omic delineation as stated in the title. So a multi-omics integration might sound very difficult, but actually I think it's just measuring how every cell responds to incoming signals. When there's a signal incoming, it induces a signaling event, often measured in phosphocytes, which we can measure. As this activation of kinases and transcription factors then leads to the production of RNA from our DNA, which we can measure in transcriptomics. And then this RNA is translated into proteins, which we can also measure in proteomics. And then finally, we have this subgroup of proteins that are excreted outside the cell, what we call secretomics. And in that way, we can, at each level of our cellular response, see and measure what is happening. So then we try to make sense of all of this data. So first we classified each phosphocyte, RNA and protein as from which response it originated. So is it either a TNF-alpha, an interferon gamma, just the addition of both, or a synergy response that is higher than each of them combined. In terms of result, we think that this synergetic state is induced somewhere in between the crosstalk of the NF-kappa-B and the JAK-STAT pathway, which are induced by TNF-alpha and the interferon gamma. But then when you combine them, there is this crosstalk that induces this synergetic state. That's at least our hypothesis. Also, what is very interesting is that the synergy is mostly visible in chemokines, so such as CXCL5 or CXCL10. Which is where the synergy is very clear. If you add one cytokine, the RNA is not made at all. If you add the other one, there's a slight increase. But if you combine them, then suddenly there's this synergetic increase. So why would you need the synergetic regulation of these specific chemokines? This is me speculating, but I think it's somewhere along the lines of the progression of inflammation or an antiviral response. A lot of these synergetic proteins are antiviral proteins. So I think when an inflammation response starts, initially TNF-alpha is released. This induces the recruitment of different immune cells, and those immune cells then might start releasing interferon gamma. And at a certain point, then you can end up with a microenvironment as your inflammation response progresses, where both TNF-alpha and interferon gamma are present. And only in that case, then these synergetic chemokines are released to attract either even more immune cells or a specific subset of immune cells that we do not know. But I think it's quite interesting that a million years of evolution have led to this very specific regulation of this synergetic chemokines that must have a role somewhere along the lines of this progression of inflammation. And that is managed in these endothelial cells. So that's a quick rundown of chapter 1, the multi-omics delineation of cytokine-induced endothelial inflammatory states. If you want to know more, you can find this paper. It's published in Communications Biology. I'll put a link somewhere. As for now, take care of yourself, take care of each other, and have a wonderful day.